Hello and welcome back to biology. Now, one thing I didn't mention last time was this. Prokaryotic cells. No nucleus. Eukaryotic cells. Nucleus. Okay, and you recall, of course, prokaryotics generally like bacteria, and there are certain differences as well um, between prokaryotic and eukaryotic cells, which I'm going to go into more detail once I explain the various organelles. And organelles, you recall, are elements of the ultrastructure of a cell. You'll also recall that in eukaryotic cells, animal and plant cells. In the first episode, I did a animal cell. Yeah. So I'm now going to draw a plant cell, and they are similar in a sense, but plant cells are more complex. So let's see how we're going to do this. I'm going to just see how we get on. Well, I'm not going to make it like that, actually. I'll do the rounded rectangle again, I think. I wonder what other shapes there are. I could actually use these lines to draw. Actually, there is an arrow, isn't there? Didn't use that last time. Didn't even think of it. I'm going to try and I don't know, try and draw things a little bit better. So I'll have a red outline. Why not have a red outline? Can make it a bit bigger. I just want to try and make it as big as I can. Yeah, that's about as big as I'm going to get without. Yeah. So I'm going to do a red outline, and then what I need to do is I need to leave some gaps for the plasmodesmata, plasmodesmata, plasmodesmata. I'm going to call it plasmodesmata. Yeah, that is not working though, is it? Well, that's one way to do it. There we go. Just put another hole there. And another hole here. Okay. Another thing to realise is that a lot of these things. Now, what we're going to recall as well is that things are slightly more complicated here. In fact, that was probably not the best way of doing things. I'm going to change to a green colour here. Let me go back through it, because what we've got is we have, of course, we have we have the sort of internal structure, but the plant cell is more complicated because it needs to transfer stuff through, like the sap, and that's the key thing. There's transport, and there's all these ideas about different types of transport. So yeah, so we're going to have the plasma, which is the cell surface, and outside, and that's actually inside the cell wall, which is going to be this outer layer here. So how am I going to do this? All right. So I'm going to do something like this. I think I'm going to need another one as well. Yep. So I might just make this red, actually, all of it red, just because it's. Well, maybe I shouldn't. Maybe I'll delineate a bit. Put some green. It's a plant after all. That is still red. Green. Alright. So this is the area we're going to have to work with. Okay, I'm going to get my eraser out, put it back to it's so strange. Okay, let's leave some gaps here. And we're going to do the gaps here. Okay, so yeah, we've got two internal layers. We've got, well, yeah. Actually, I'm going to actually fill it with a lighter colour first of all. Because this way, I'll just fill it with this colour. Now I can do that. So yeah, so we're going to have... Okay. We're once again going to... There's not really a better way to do this. We need to make sure these channels are open. We're ready for nutrient transport. Alright, so basically what's going to be happening is this outer layer is going to be the cell wall, and then the inner layer is the cell surface, the plasma, and then of course inside is the cytoplasm once again. So now, and yeah, and these are actually you know, the plasma and the cell wall organelles. And like I say, in the next episode, I'll be speaking about the organelles. So now, I'm going to draw the new structures first, because this will help to, you know, do some isolate these things. So yeah. So this is the vacuole. This contains the sap. It's generally a collection of minerals and nutrients. There's the vacuole. Now I need to put in chloroplasts. So again, I'm going to try to do make this look like a, yes, not bad. To do it like that. Change the stroke size to something a bit smaller. Yep. These are going to be chloroplasts. Of course, it's to do with photosynthesis, which I'm sure we'll be talking about later on down the line. Okay. From there, we're looking at fairly similar things, so it might be a good idea to start. I'm trying to remember how I did this last time. Let's start with the nucleolus. So I'm going to try it a bit differently. I'm going to try and get this as close to the center as I can. Just you know, give it a bit more. I think last time I actually did it as a circle. Just make it a bit try and make it a bit bittier. And then yeah, and then we've got the cell envelope. So let's just do it. I'll try and make it a bit smaller this time. So I'll put all the bits in here. Switch to yellow. What are we on with regards to? Yeah, that one I'll do. Switch everything to yellow. Let's get this. No, I don't like it like that. There we go. I'm going to change the stroke size to a bit smaller. Well, this is pretty small. I don't know why the stroke size doesn't really change much. Actually, you can look at brushes as well. Oh, yeah, maybe I was using the wrong brush. Probably could use a pencil. But it's good. It's good to look at all the menu options and stuff. All right. So yeah, I think you can tell there's a bigger structure there. So I'm going to do the envelope now. Okie dokie. Yeah. Of course, there's lots of different ways that these things travel around. And all these things can look different, even though we've got the same things. So there we go, slightly different looking from last time. But then I suppose now what we've got, mitochondrion. Now the thing about the mitochondrion is they can fit in anywhere. Ribosomes are little red dots. Do a few of those. Alright, there's some ribosomes. Now what about Golgi apparatus? Golgi apparatus, rather. Yeah, just say that. No, it is Golgi. Or is it Golgi? Well, I'm going to find out for that. Is it Golgi or Golgi? That's not that important though, really. I mean, well, obviously it is, but not at the moment. So that's going to be, do that yellow as well. And yeah, I need to go much. I need to change this brush. I want to use an actual calligraphy brush. You know what? I might just be better just using a pencil. Yeah. And the structure of this is a bit like, I don't want to click these silly buttons on the pen. Oh wow, it doesn't come through very strongly. No, that's unacceptable. There we go. Kind of a big girl fountain pen. Well, that's not a fountain pen, but it looks better. I'll draw like that. Yeah, that looks alright. Mitochondrion, I don't need to draw. We've got the rough and the smooth endoplasmic reticulums. Let's draw them somewhere. So there's some nice smooth ones. There's some rough ones. Oh god, I've got the nucleus, nucleolus, SER, RER, chloroplasts, vacuole, ribosomes there. Okay. Time for some mitochondrion. So it's best for last. Okay. Let's get some nice mitochondria on the go. I wonder if I put little black dots in them before. So these are... This is sort of... Like, oh wow, that mitochondria didn't turn out too well. Give them a little... And now I'm going to be really... Just going to try and do a little detail on the mitochondrion here. Nope, nope, nope. So just do... I actually feel like I could probably get slightly more, yeah. Interesting. I can actually get some nice... Alright, so now comes the fun part, the labelling. Now. This may end up being absolutely massive though. No, it's not. I don't want to do that though. What colours should I make the arrows? We've got blue. Yes. Could actually I could custom colours as well. And I wonder I could even put a text thing in there. How would I go about doing that? I wonder. No, I don't want to do that. I can't get out of it now. Look at this new tab's opened. Let's get out of it. I'll get out of it. There we go. Forget about that. Alright. So, chloroplast time. I don't really like this arrow, to be honest. I prefer just drawing an arrow. How am I going to get rid of this arrow now? With difficulty from the looks of it. There we go. Alright, let's just get into this now. Not like that. Pencil. Alright, here we go. Chloroplast. So it's referring to this layer here. Okay. And this is the rough endoplasmic reticulum. And these holes here. Plasmo. Wow, interesting there. And then, old favourites from animal cells. Mitochondria. Oh, I've made that a little bit too big there. Well, this is the vacuole. I'm going to just actually use the bottom. No, you know what? I may as well just. I could probably just get rid of. I don't know. Fuck it. I'll just go over it. And this is the Golgi. I'm just gonna put Golgi because I'm not sure about this. It might be. All right. So this is smooth endoplasmic reticulum. This is cytoplasm. And just go through the mitochondrion straight to the nucleolus. And there's the nucleus there. Yeah, so I've done the I've done most of it. So again, ribosomes. And cell wall. So yeah. So yeah, I've managed to pretty much put that on the page, just about. I could actually... no, you can't really change it much. I don't know what some of these other buttons do, but anyway. So yeah, and like I say, these things are only about between 2 and 200 micrometers. So a micrometer, I believe it's a millionth of a meter. So yeah, I'm just going to talk a little bit about that now. It's a millionth of a meter. So that equates to 0.001 millimeters. And eukaryotic cells can be between 0.002 millimeters to 0.2 millimeters. So they can get, 0.2 millimeters. It's not very small really. It's almost visible to the human eye. Of course, there's various things, microscopes and things. I believe, one final thing I just want to do is actually I'm not going to do it quite yet. I have a feeling it's the bottom one here though, the Golgi apparatus. So yeah, so what I'm going to do next time anyway is I'm going to talk in more detail about all these elements. Um, Obviously, there's more for this. Oh yeah, let's just label this. Oh wow, look at the size of that. Oh wow. This is perhaps not the best pen to use. Plants are. Well, certainly some interesting pens there. Um, and with this one, it actually, but you know what? That's not hugely relevant to biology.